Chloroplasts, an organelle that is specialized and found mainly in the plant kingdom. And whenever one thinks about chloroplast, one thinks of photosynthesis. So let us look at the learning outcomes of this session. Chloroplasts are organelles that are present in plant cells and algae, and they are specialized, as mentioned, in the process of photosynthesis. Like mitochondria, chloroplasts too have more than one membrane and a system that enables transfer of electrons and protons to enable production of ATP and NADPH2. Now, this process in itself in the chloroplast requires light. Using the ATP and NADPH2 that is formed uh, due to the above mentioned step, the chloroplast stroma has the molecules of Calvin cycle that fixes carbon dioxide to produce sugars. Not just sugars, you can also have formation of amino acids and fatty acids. This process does not require light. Chloroplast has its own DNA and genetics, its own ribosomes and RNA for protein synthesis. Now, when one looks at the structure of chloroplasts and under the microscope, especially the light microscope, chloroplasts are generally visible as green bodies of a few micrometer in size. So, as you can see over here, chloroplasts are very clearly pigmentous, and the pigment that is prevalent is the green color, which is attributed to the presence of chlorophyll. But it also has shades of yellow because of the presence of other chromogenic substances. A higher resolution is actually revealing the fact that the chloroplast has a very compartmentalized structure, and it has very specific regions which are called as grana and stroma. So this is what is called as a stroma, and this is what is called as grana. And in fact, the grana and the membranous structure that is present in the stroma is known as thylakoids. And it is these thylakoids that have or that contain within it the chlorophyll, which is the pigment that is responsible for green coloration of the chloroplasts. The name chloroplast also is thanks to the fact that it has chlorophyll. Now let us compare chloroplast with mitochondria. Why we compare the two is because both chloroplasts and mitochondria enable the formation of ATP through chemiosmotic hypothesis. So in size, chloroplast is much larger than the mitochondria. Like mitochondria, the chloroplast also has an outer membrane and an inner membrane. However, the inner membrane of the mitochondria is highly folded in structure, whereas the inner membrane of the chloroplast is not folded. The outer membrane of both mitochondria and the chloroplast is much more permeable than the inner membrane, and the inner membrane in itself has very specific proteins that allow only specific components to enter what is termed as the matrix in the mitochondria and the stroma in the chloroplast. So this is stroma, and this that you see in the mitochondria is the matrix. Now, interestingly, what has been observed is that the chloroplast has a separate third membranous system which is folded, and the folding is such that they are present as stacks of or disc-like sacs, and these disc-like sacs are flattened and they exist one over the other as saucers, cup and of the cup saucer, the saucers one over the other. And each of these sac has within it a lumen. So these flattened sacs are what are called as thylakoids, and every thylakoid would have a thylakoid lumen present within it because there's space inside this membranous system. Now it is this third membranous system that is present that is allowing what is referred to as photosynthesis. So it can capture light and convert it to energy. But interestingly, what is observed is that the thylakoid is present as a stacked form, and the stacked form is what is called as the grana, and it also has non-unstacked forms of thylakoids which is present in the stroma. So therefore, this differentiation into grana and the stacked thylakoids and the unstacked thylakoids also has to do with the kind of proteins that are present in the membranes of the grana and that in the stroma. They are different, and so there are only certain functions that happen in the grana and certain functions that would happen in the unstacked thylakoids. So therefore, chloroplast is an organelle like mitochondria which is involved in energy conversion, and chloroplast is mainly associated with, with converting light energy into chemical energy. So effectively, when one observes the photosystems that are present in the thylakoid membrane are able to absorb light and they're able to split water. So oxidation of two water molecules by a photochemical reaction, this is in fact the reaction center, and it produces one molecule of oxygen. As you can see, the oxygen is moving out of the chloroplast and it is released into the atmosphere. So plants are known to absorb carbon dioxide and give out oxygen thanks to this process. Now, why is there a need to split the water molecules? The need to split water molecules is to actually get electrons, and these electrons that are withdrawn from the water are transferred to a second type of photochemical reaction, uh, which was going to then produce NADPH from NADP+. And in the process of transferring electron from one photochemical reaction center to another photochemical reaction center, a proton gradient is formed, and that proton gradient enables the formation of an ATP molecule as well. These two things we are going to look into slightly more detail in the next few slides. Now, once you have the ATP and the NADPH formed by this reaction, these sets of reactions, the NADPH and ATP are utilized by another cycle in the stroma, which is called as the carbon fixation cycle. This is where carbon dioxide is taken up and it is fixed to form sugars, amino acids, fatty acids. Now, interestingly, therefore, in the chloroplast, you have distinctively the photosynthesis divided into what is called as light reaction and the dark reaction. So the light reaction is where the light is absorbed and the energy is converted to ATP, which is a chemical energy, and also one gets reducing equivalents in the form of NADPH. Now, once you have these two forms, so these two are products of the light reaction. The products of this light reaction in the stroma is able to be used up to form the organic compounds. And therefore, this process, this Calvin cycle of carbon fixation does not require light, and hence it is called as dark reactions. So, this in fact seems like a misnomer. It does not mean that you cannot have Calvin cycle happening during the daytime when there is excessive light. It just means that this reaction or this set of reactions does not require light, while this set of reaction requires light. So that is why this set of reactions falls under what is called as light reaction, and this set of reactions is what is falling under dark reactions. Now let us look into just a little detail on carbon fixation. This, as mentioned, is carried out by Calvin cycle. So the Calvin cycle per se is divided into three major parts. One is what is called as carboxylation, the second part is what is called as reduction, and third part is what is called as regeneration. One of the major enzymes that is involved in the Calvin cycle is the rubisco. And rubisco is very characteristic of chloroplasts. In fact, rubisco has two subunits, and the larger subunit of the rubisco is formed inside the chloroplast itself. So the rubisco is actually able to, so the full form of rubisco is ribulose bisphosphate carboxylase and oxygenase. So it can act both as a carboxylase as well as oxygenase. So when the oxygenase is active, then it carries out what is called as photorespiration, while when the carboxylase is active, it carries out carbon dioxide fixation. So as you can see over here, carbon dioxide is accepted and transferred to ribulose 1,5-bisphosphate, and that forms a intermediate which is a 7-carbon structure, which is then ultimately broken down into a 3-carbon component, which is 3-phosphoglycerate. Now this 3-phosphoglycerate, by a set of two reactions, is then converted to glyceraldehyde 3-phosphate, which is also a 3-carbon structure. This process requires ATP and it requires NADPH. So all the ATP and the NADPH that is formed by the light reaction is used up by the Calvin cycle. So therefore this dark reaction requires the ATP and the NADPH too. This glyceraldehyde 3-phosphate, one molecule of which is siphoned off, and that this siphoned glyceraldehyde 3-phosphate acts as a precursor in the stroma to form several different organic compounds like sugars, fats, amino acids. The other 5, glyceraldehyde 3-phosphate, through a set of reactions, again gives back rise to ribulose 1,5-bisphosphate, which can again interact with or react with carbon dioxide, and this cycle then ultimately goes on. So therefore, it is interesting to note that for every 3 molecules of carbon dioxide that enters the cycle, 1 molecule of glyceraldehyde 3-phosphate is produced and 9 molecules of ATP and 6 molecules of NADPH are consumed. So this is the stoichiometry of the Calvin's cycle. Now, therefore, between the chloroplast and the mitochondria in plant cells, in algae, etc., what is ensured is that the cell is getting a constant source of metabolites and also ATP. So the chloroplast is able to give rise to sugars, etc., and which in turn are further metabolized to get different metabolites, while sugars can also go to the mitochondria and there, due to oxidative phosphorylation, give rise to ATP that can be used by energy. So between the two of them, they are able to ensure that the cell has enough of metabolites as well as ATP. Now let us look at the photosystems, which is taking care of the light reaction in the chloroplasts. And these photosystems are lodged, or they are basically residing in the thylakoid, which is the third membranous system in the chloroplasts. So as you can see, this is the thylakoids, and in the thylakoids, you have photosystems present, and a photosystem characteristically has a reaction center, and this reaction center has a special pair of chlorophyll, and this special pair of chlorophyll is able to accept electrons and get excited, and therefore there has to be a sufficient amount of energy obtained that would enable this special pair of chlorophyll to get excited to a higher state. So in order to have that amount of energy required for the electrons to get excited, what is observed is that near the or surrounding the reaction center are certain complexes called as antenna complexes, and these antenna complexes have pigments other than chlorophyll. So those pigments are also able to absorb light of different energies. Therefore, they will all collect the light of different energies and then gradually bring all the light to the reaction center. So all the light that comes up is necessary ultimately for the reaction center to get active. So this is something that is considered to be therefore a photosystem. So a photosystem has the reaction center and it has the light harvesting antenna complexes. Now in chloroplast, in the thylakoid membrane is present two photosystems, one photosystem 2 reaction center and a photosystem 1 reaction center. And together these two photosynthesis systems form what is called as the Z scheme of for photosynthesis. So as you can see, The photosystem 2 is able to absorb energy. The energy and the electrons and the is transferred because the water is split to give rise to electrons. That is carried to the photosystem 1, and then you have this photosystem 1 enabling formation of NADPH. So therefore, you need photosystem 1 as well as photosystem 2 to carry out photosynthesis, and because it looks like a Z, it is called as the Z scheme. Photosystem 1 is in the unstacked thylakoids of the stroma, while photosystem 2 is present in the stacked thylakoids of the grana. So you can see that photosynthesis itself, you have the locations of the two photosystems at different parts of the thylakoids. Let us go through the Z scheme in brief. So the Z scheme has, as you can see, this is the photosystem 2. The photosystem 2 has a complex which has a lot of manganese that can split water. So manganese ions are present and they can split water to give rise to oxygen and also give rise to electrons and photons and protons. And these electrons are then through photosystem 2 getting excited. Now you can see how it goes from one redox potential to another redox potential. Now when you have the electrons getting excited, these electrons can always fall back. But instead what happens is it is accepted by another complex which is able to accept electrons from photosystem 2. So this is what is plastoquinone. Plastoquinone is able to transfer its electrons to the cytochrome B65 complex. The cytochrome B65 complex is able to pass it on to plastocyanin. So therefore, the cytochrome B65 complex in fact is considered to be a connecting point between photosystem 2 and photosystem 1. The electrons 1 transferred to the photosystem 1, okay, these for these electrons are then accepted by ferredoxin, and from ferredoxin the electrons are by ferredoxin NADP reductase used up to convert NADP to NADPH2. So therefore, one of the products of this transfer of electrons and protons is the NADPH, which is a reducing equivalent that is required for Calvin cycle. So you can see how this light reaction is essentially giving rise to a product that is required by the dark reaction. But in the process of the electrons getting transferred from one system to the other system and finally getting accepted by NADP, what is also observed is that the cytochrome B65 complex is able to push out protons into the thylakoid lumen, and that pushing out of the protons through the thylakoid lumen to the thylakoid lumen leads to what is called as the proton gradient, and that proton gradient is used by ATP synthase to synthesize ATP. So to just understand that again, so as you can see, light is accepted by photosystem 2, photosystem, and at the same time you have a water splitting enzyme using manganese ions. They convert or they split water such that electrons and H plus are obtained and oxygen is released. These electrons and protons go to the plastoquinone. From plastoquinone, they move to the cytochrome B65 complex. From this into plastocyanin. In the process of this electron transfer, there are several protons that are pushed out through the cytochrome B65 complex. So this stroma is from where the protons are pushed out or pumped out into the thylakoid space. So therefore what is going to happen is that the thylakoid space will have more of protons, leading to formation of what is called as proton gradient across the thylakoid membrane. Now this proton gradient that is formed is going to give rise to proton motive force. At a certain point, that force is going to activate the FOF1 ATPase, which is also called as the ATP synthase. So the ATP synthase will allow the protons to move in, and in the process of allowing the protons to move into the stroma again, the ATP synthase is getting activated and it converts ADP into ATP. So at one end, with the transfer of electrons, the final electron acceptor, the NADP plus, is getting converted to NADPH, but due to this process, you have the proton gradient formed, and that proton gradient allows the formation of ATP. So this is what the photosystems are doing. Photosystem 2 and photosystem 1 are enabling formation of NADPH and ATP. And the ATP production is because of the electrochemical proton gradient, which is basically what we understand by chemi-osmotic hypothesis. So you have coupling of electron transfer with phosphorylation, and therefore that is how the ATP is getting synthesized. Now, the chloroplasts have their own genomes. So, what has been very distinctively studied is that the DNA that is present in the chloroplast is a closed circular covalent DNA, and this closed circular covalent DNA is very similar to the DNA that is present in bacteria. So, this is one thing that has been studied over a period of time, and the presence of a closed circular DNA within the chloroplast is suggestive of the fact that chloroplast has been obtained from a bacteria. So the chloroplasts seem to have evolved from a bacteria. This is something that the endosymbiotic theory has proposed, and there are a lot of facts and there are a lot of things that actually show similarities between chloroplast and bacteria. As you can see, the chloroplast genome has several key genes for tRNA, for ribosomal proteins, and one must understand that these tRNA and ribosomal proteins are able to translate and produce proteins within the chloroplast. So in fact, you can see that ribulose bisphosphate carboxylase, the large subunit, is because of the gene that is present in the chloroplast DNA and not into the nuclear DNA and not the nuclear DNA. Although what is understood is that many of the genes from the chloroplast DNA have been exported to the nuclear DNA, and so the chloroplast cannot function solely based on its own DNA. It also needs several proteins from the nuclear DNA, and therefore there is a lot of proteins that enter the chloroplast through very specific protein receptors or through very specific protein transporters. So that is something that is very similar to mitochondria as well. So you have a lot of important genes. In fact, ATP synthase gene is also very important. You have genes also for the B6F complex. So all these enable the chloroplast to function properly. Interestingly, chloroplast can also divide by binary fission. This is something that is very similar to how bacteria divide, and so that is again another resemblance between a bacteria and a chloroplast. So, effectively, this endosymbiotic theory suggests that or proposes that a eukaryote, a primitive eukaryote or a primitive aerobe must have engulfed a cyanobacteria, because the photosystems that are present in the chloroplast are very similar to the photosystems that are present in the cyanobacteria. So the chloroplasts have their own system for transcription and translation, and one another important connection between the bacteria and the chloroplast is that the ribosomes that are present in the chloroplast are the 70S type. Also, the chloroplast DNA is reported to show bipaternal inheritance. That means you can get the chloroplast DNA from either father or mother, although the maternal inheritance is more pronounced than the paternal inheritance. And interestingly, defects in the chloroplast DNA or in the chloroplast itself also leads to several changes both phenotypically as well as genotypically in plants. So let's say, for example, the money plant, the common money plant or the pothos. When you see the leaves of money plant, you will find that the leaves are variegated. Certain portions are white, certain portions are green. So those portions that are green have chloroplasts with a large amount of chlorophyll, while those which are white in the pattern or the variegation is white, it means that those chloroplasts are not colored. So thereby it has been referred to as a defect in the chloroplast itself. So let us make the conclusions. Chloroplasts, proposed to have evolved from cyanobacteria, are a specialized organelle with its own genome in plant systems, with photosynthesis being its main function. The chloroplasts have a third membranous system which holds the entire complexes of photosystem 1, 2, and ATP synthase, mediating absorption of light and converting it to chemical energy. Both photosystems 1 and 2 are able to carry out redox reactions to transfer electrons from water to NADP to produce oxygen, NADPH2 and ATP using the generated proton motive force. The carbon dioxide is then fixed in the stroma using the Calvin cycle to produce sugars, fatty acids, etc. So chloroplast structures have evolved to carry out the light reactions and the dark reactions to synthesize organic molecules from carbon dioxide and light. These organic molecules are produced virtually for all living organisms. Thank you.